Good morning, good afternoon, good evening, wherever you're joining us from in the world today. It brings me great pleasure to welcome you all to our 5th annual Targeted Protein Degradation and Induced Proximity Symposium. My name is Joel Crisser-Brown. I'm a product manager in the innovation team here at Bio-Techne, and I'm excited to be hosting today's event. For today's symposium, I'll start out with a short introduction and some housekeeping items. Then we'll kick off the series of presentations. Each talk will be followed by live Q&A, so please send your questions in throughout. We have lots to get through, so let's get started. This event is brought to you by Bio-Techne. Bio-Techne is a global developer, manufacturer and supplier of high-quality reagents, analytical instruments, and precision diagnostics. Our brands include R&D Systems, Novus Biologicals, Tocris Bioscience, Protein Simple, ACD, Exosome Diagnostics, Assuragen, and Lunaphore, to name a few. We have 34 locations worldwide with over 3,000 employees and offer more than 500,000 products backed by over 350,000 citations. So over our many decades of developing reagents and technologies for life science research, we've carefully curated a comprehensive toolbox of reagents to reduce bottlenecks in TPD projects across the following areas. And these can be explored via our dedicated TPD page, to which there is a link to in the chat. So when starting a TPD project, it's important to understand a biological pathway of interest and validate your targets for degradation. Known targets can be degraded using our extensive range of tool degraders and molecular glues licensed from academic institutes and biopharma, or developed here in-house and synthesized at our chemistry facility in Bristol in the UK, while degradation can be quantified using our extensive catalogue of trusted antibodies. For generalizable methods to degrade new targets, we offer an extensive range of reagents for Degron tag assay development, including fluorogenic probes for imaging tag-labeled proteins, as can be seen in some new data with DTAG Genelia Floor on the screen here. For degraded design and synthesis, we pioneered a range of building blocks for onward chemistry consisting of E3 ligase, LITAC, and induced proximity ligands. These building blocks can be incorporated into high-throughput chemistry platforms to reduce the medicinal chemistry bottleneck of your TPD projects. Through a dedicated team in Minneapolis, we ease the burden of expressing and purifying bulk quantities of highly active E3 ligase proteins through our catalogue of high-quality recombinant proteins and antibodies suitable for in vitro assay development. Robust assays are crucial to the success of any TPD project, and employing a wide array of orthogonal assays will accelerate the generation of vital data to move forward. For target engagement and ternary complex formation assays, we recently launched a TR-FRET toolbox, including the next generation CoraFloor TR-FRET donor developed by a fantastic team at Harvard University who presented on this at last year's TPD Symposium. CoraFloor offers enhanced TR-FRET assay performance at significantly reduced cost per well for your screening campaigns. We have off-the-shelf reagents and proteins for in vitro ubiquitination assays, automated fully quantitative Western blotting with Simple Western, or as many of you may be more familiar with, our JESS instrument, which I'll speak more about in a few slides. A comprehensive suite of downstream assays to probe the pharmacological effect, from cell viability and DNA damage assays to antibody arrays and RNA-scope in situ hybridization assays. As induced proximity research trends towards an increasing number of non-degrading modalities, so does the requirement for complementary assays to quantify these interactions. This year, we're proud to launch the Molboolean assay kit, which is a state-of-the-art all-in-one solution for analyzing protein proximity within cells and tissues. With this kit and your own user-selective mouse and rabbit primary antibodies, you can simultaneously detect A, B, and AB interactions in a spatial context to further quantify protein-protein interactions in your induced proximity discovery projects. Please check out the attached app note on the GoToWebinar screen for more information. Simple Western technology is an automated capillary Western system that uses capillary electrophoresis for protein separation, followed by immunoassay-based detection within a fully enclosed system. Here's an example of rapid, reproducible, and quantitative dose response data generated on a Simple Western JESS instrument using a BTK degrader available from Tocris or Bio-Techne and the parent inhibitor ibrutinib, as well as several controls. Simple Western technology reduces the time to fully analyze results in just 3 hours, provides absolute and relative protein quantification, is highly reproducible, and uses a very low sample volume, making your precious samples go further. All this enables you to quickly and confidently characterize the efficacy of your degraders to obtain reliable DC50 values. And the Simple Western family is growing. We have a really exciting announcement coming on July 31st. We're still a few weeks away, but here is a little teaser video that we'd like to show you ahead of that. So with that, I'd like to introduce our first speaker today, Jolene Poisson. Jolene received her bachelor's degree in cellular biology and physiology, followed by a master's degree in microbiology with a specialization in virology from the University of Strasbourg, France. She completed her PhD on the hijacking of the ubiquitin proteasome system by HPV at the University of Strasbourg, France. And in 2017, she joined Dr. Taipali's lab at the Donnelly Center, University of Toronto, as a postdoctoral fellow. She's currently senior scientist at the Induced Therapeutics, where she works on targeted protein degradation. So before I hand it over, just a quick reminder that we'll take any questions following each presentation. So please submit your questions as they come to you, and we will respond following the presentation in the Q&A session. Okay, over to you, Jolene. Thank you.
Thank you for the very kind introduction. So today I'm going to talk about what happens when you force protein to interact with one another. So nature has evolved elegant mechanism to regulate the physical distance between molecules. We call that also proximity, for a wide variety of purposes, whether it is activation of cell membrane receptors, neuronal transmission across the synapse, or chrome sensing in bacterial biofilms. Proximity is a ubiquitous regulatory mechanism in biology. Many pathogens, including viruses, bacteria, or even plants, through hormones, are regulating proximity. A very famous example is the E6 protein from HPV that is capable to hijack E6AP and E3 ubiquitin ligase to degrade P53. P53 will never be degraded by E6AP without the oncoprotein from HPV. So modulating proximity with small molecules is one of the most promising innovations in drug discovery. But the main question is, how do we pair a target on one side and the effector on the other side? We have plenty of protein in the air film. How do we identify the one that can lead to degradation or to phosphorylation or transcription activation if we're interested by other mechanisms? So this is the protein pair problem. Many E3s are identified already in the air film, in the proteome, but only a few are actually used in targeted protein degradation, mostly cerebron and VHL. So the idea in my work was to develop a method that can identify such effector protein in a scalable and unbiased manner. So we established a synthetic proteome scale platform to functionally identify human protein that can promote the degradation or the stabilization, on the other hand, of a non-native substrate protein in a proximity-dependent manner. So this platform comes in two flavors. One is inducible and the other one is constitutive. The principle is quite simple. We have a sensor, which consists of an EGFP molecule fused to ABI1, followed by an IRS, and then tag BFP. And we express the effector. So this is the OR fused to PIL1 or VHA-GFP, which is a nanobody that recognizes GFP. In the case of the constitutive platform, we have the nanobody recognizing GFP, which bring the protein of interest close to the substrate that can lead to degradation or to stabilization in another end. For the induced interaction, we use abscisic acid to bring PIL1 and ABI1 close to another, which reconstitute the system. And because the EGFP is fluorescent, it makes the system very easy because we can follow up the degradation or the stabilization using flow cytometry, for example. So the first thing we did was to look at known degraders. So we look at cerebrand and also SPOP, either the wild-type protein or the one deleted from the math domain, which is substrate recognition domain. And what you can see here, you have EGFP and tag BFP is after addition of abscisic acid for SPOP. We see a shift down, suggesting that there is less GFP, whereas BFP is stable. So this is a clear degradation of the substrate. It's even better when you remove the math domain, and this is not as good for cerebrand, but cerebrand in the system wasn't expressed really well, so it might explain why. We observed the exact same phenomenon with the constitutive system, with the VHH-GFP. And in this case, the negative control is nanolog. And you can see that we don't see any degradation, maybe a bit of stabilization in this case. So the systems seem to work. So then the idea was to move to a screen to identify new potential degrader or stabilizers. So what we did is we created a cell line that was expressing the sensor, so EGFP, ABR1, and BFP. And we infected it with two different libraries, either the Orpheum fused to PIL1 or to VHH-GFP. After selection, we use flow cytometry to sort the top 10 population for stabilizer and the bottom 10 for degrader. We extracted the genomic DNA, sent everything for sequencing, and this is the results we obtained. So this is the degrader side, so the decrease in GFP that I'm showing you first here. And this is a volcano plot, so you have the p-value and here the fold change. And what you can see is we identified quite a lot of genes that were enriched in this population, and I highlighted in orange all the ones that were affiliated to the ubiquitin proteasome system or autophagy or even proteases. And a lot of them were. You can see a lot of ubiquitin ligases, which is expected. So we were quite happy at first because it suggested that the system was working. But we also identify proteins that were new and not related to the ubiquitin proteasome system. So that was also quite interesting. Something we did right after is to look at E3 by themselves. So we rearray all the E3 reads. So we tested a lot of them, almost 300 of them. And we use the constitutive system. So we fuse every E3 ligases to the VHH-GFP nanobody. And we just look at EGFP degradation, same system. So this is a very busy slide, so I'm not expecting you to read every E3 names, but very quickly, when you have degradation, you look at the blue color, and when you have stabilization, this is red. So they're all organized by family, and what you can quickly see is that one family doesn't necessarily lead to degradation or stabilization, suggesting that a domain organization or a type of domain is not enough to explain the behavior of an E3. So you can't really predict that this family or this specific E3 will lead to degradation in our system or to stabilization in another case. This is even better highlighted if you look at the FBOX family right here. So FBXL15, for example, is a very good degrader in our hand, and FBXL8, which is closely related to it, in this case would act mostly as a stabilizer. So this is the guilt by function or guilt by association issue. So suggesting that you need to try on each E3 instead of assuming which role they will have. UB2B, which is an E2, so an E2 conjugating enzyme, was also one of the top ist in our degrader screens. It was a very, very strong and potent degrader. So we decided to look a little bit more at it. So the first thing we did is we mutated the catalytic domain of UB2B just to see if the E2 function was necessary for its function. And this is what you can see right here. Once you mute the catalytic domain, you don't get any degradation anymore. Because it's an E2, it's also known to interact with E3 ubiquitin ligases. So in this case, RAD18 and RNF20. So we also mutated the 2 pockets that make contact with the E3, just to see if you have to bring in E3 to be able to degrade the target. And in this case, you can see that we obtain degradation with all the mutation, suggesting that UB2B by itself is enough to induce degradation of EGFP. And we know that this degradation occurs through ubiquitination because we got ubiquitination of the substrate with the wild type UB2B, but not with the catalytically dead mutant. We also assessed almost all the other E2. The E2 are a very close family. They are all very highly similar to one another, and UB2B was the best one, along with UB2A, that is the closest member among the E2. And we also found the UB2D family, which matches a recent development of molecular glues for this family. So UB2B was quite unexpected because it's an E2, but this one seems to be a pretty good degrader in our end, in this system at least. What about stabilization? So far I focused on degradation, but we also identified some potential stabilizer. So in blue this time I highlight known regulators of protein stability. So you can see some chaperones, some deubiquitinases, and also protein involved in protein stability in general. But we also found a lot of protein that were involved in protein stability and mostly in protein metabolism. So we think that it is possible that stabilizer act both catalytically and non-catalytically. Catalytically would be through removing ubiquitin, such as the ubiquitinase, for example, or modulating other post-translational modification. And non-catalytically, they might shield the substrate from ubiquitination, sequester it to specific cellular compartment, or just simply increase the thermodynamic stability of EGFP. And this is what you can see here. Stabilization screen hits were themselves significantly more stable than the rest of the orpheome. So you can see the increase. And in contrast, the degrader hits weren't more stable or less stable. So it seems to be through the mechanism seems to be more specific in this case. Similarly to what we did with the E3 ubiquitin ligases, we look at DUBs. So because we were looking for stabilization, this time we changed a little bit the sensor. So we used a mutant of GNMT, which is known to be very unstable. We fuse it to EGFP, and then we have DS-Red as a sensor control right there. And all the DUBs were fused to DHA-GFP. And then we looked at stabilization. So in this case, white is no stabilization, and dark red, this is stabilization of the sensor right here. And if most of the DUBs can induce at least a little bit of stabilization, only a few of them were capable to really strongly stabilize the sensor. And among them, we have OTUB1 that is already used for some stabilization in an induced proximity manner. And we know, for example, for some of them that the function of the DUB is necessary because when you mutate USP38 in the UCH domain, you don't get any stabilization anymore. So this stabilization seems to occur through deubiquitination of the substrate. Another very interesting hit we got on the stabilization side was KLHL40, which is an E3 ubiquitin ligase. It belongs to the BTB-Kelch family. And initially, you would expect this one to be a degrader, but in our end, it was a very strong and robust stabilizer. And something to mention is the motif in the BTB domain that is supposed to recognize the BTB is mutated in KLHL40, so that might explain why this one is not acting as a normal E3. So something we did is we mutated and we removed the different domains of KLHL40 just to understand which part was important for the stabilization. And as you can see right here, the BTB domain and to some extent the back domain were the one responsible of the stabilization. It is also something you can see on the immunofluorescence here. This is the KLHL40 fused to the VHH GFP. When you remove the BTB, you can barely see GFP, same for the back domain. The KLH domain seems to not be important in that, but might be important in the solubility because we saw some aggregation. And this effect is proximity dependent because when you fuse KLHL40 to another tag or to no tag, you don't get any stabilization. You need the nanobody to bring KLH40 to the target. Then something else I want to talk about is to characterize the mode of action of these effectors. So to do that, we selected a panel of about 50 different effectors, mostly degraders, but some were also stabilizers, and they belong in different class of family. So some of them are related to autophagy, other ones are E3, and other are involved in other processes, like the GPI one here that were significantly enriched in the degrader screen, and also other proteins that weren't really related to anything degradation related. So the principle is quite similar. We have the same sensor, a GFP-ABI1, and we express the effector fused to VHH GFP. And as you can see here, when it's blue, it's degradation of EGFP, and red, stabilization. And the additional panel under, the color are a bit different, but basically, when it's orange, it means you don't see the previous effect, so you have a rescue. So for example, the two first line, the first one is a proteasome inhibitor, and the second one is the E1 inhibitor. So when you inhibit both the proteasome or E1 activation, basically you don't get any degradation with most of the effectors, which kind of validate the results. And we also use other inhibitors like cycloximide, which is a translation inhibitor. We have CB5083, which is a VCP inhibitor. The next one is an autophagy kinase ALK1 and ALK2 inhibitor. And the last one is an edylation activation enzyme inhibitor. So the effector reacted differently to these inhibitors. And something to mention, for example, is the last one, the edylation inhibitor. When you prevent the formation of the CRL, basically you rescue the phenotype for most of the E3. Not all of them, but some are already identified and known in the literature to be able to ubiquitinate the substrate without nedylation. And so far everything was done with EGFP-ABI1, so that's the only substrate we tested. So we decided to build other substrates, though they are all fused to EGFP, but they are fused to many different proteins that are known to localize in different compartments in the cells. So we wanted to see if a different substrate and also a different location would have an impact on the degradation. And we assessed the same 50-ish effectors on that. So again, when it's blue, it means you see degradation, a loss of EGFP, and when it's red, it's an increase in EGFP signal. And so if we focus first on this group of protein, for example, this is the one I will call angry E3. Whatever the target is, they are going to degrade it. They don't really care. They are very robust degrader. And the compartment doesn't seem to affect them too much. Same thing for these two effectors, for example, in the opposite way. They are very good stabilizer. And among them, you can see KLHL40. In another hand, you have some that are very specific, like ATG14. That seems to be able to degrade different targets, but only if they localize to the plasma membrane. So localization in this case seems to be really important. And then you have groups of effectors that are pretty robust degrader, but for only a few targets, and they don't necessarily localize to the same place. So if we take VHL, for example, this is a pretty robust degrader of our original sensor, and also to histone or to LAMP1, which all localize in different compartments. So this is not easy. It could be tricky to predict the degradation of a specific target with some effectors, because it is important to pair the proper effector to the proper target. Because you can see also that some target, for example, will be degraded by many effectors, like EGFP-ABI1, and some other, like histone, for example, will only be degraded by a few of them. So there is a specificity. So far, everything was done in fusion with EGFP. So this is good. We can degrade or stabilize EGFP, which could be a pretty good tool in a lab, but in practice, it's not that interesting. So we move on from EGFP to a more endogenous target. And one of them was this BCR-IBL fusion that is known to be recognized by this monobody that will recognize the HA2 domain of the fusion. So we made stable cell line in K562 cells expressing the effector fused to the nanobody, and everything is state inducible. And then we look at degradation of BCR-ABL. And some of our very hungry E3, if you remember from the previous slide, FBXL 12 and 15, for example, are capable to induce a very strong degradation of BCR-ABL, way better than Cerebron, for example. And because BCR-ABL is also necessary for the cells, we looked at relative proliferation of the cells after induction of the effectors. And granular luciferase was an inert protein in this system, was used as a negative control, and you can see that expressing Cerebron doesn't seem to reduce the proliferation, whereas FBXL 12 and 15, both of them strongly reduce the proliferation of the cells, suggesting a very efficient degradation of BCR-ABL. So we use exactly the same cell line to make denograft in mouse. So once again, we have the effector fused to the monobody, and everything is state inducible. So we injected the cells into the mouse. About 2 weeks later, once the tumor was detectable and big enough, we induced DOX, or vehicle control. And we look at the size, the tumor volume, the volume of the tumor. And for Cerebron, for example, it didn't seem to impact too much the volume across time, but for FBXL 12 and FBXL 15, we saw a reduce in the tumor growth, or even an arrest of it for FBXL 15. So we were quite happy, and it's showing that some of the effector that we identify from a screen with a very artificial method, which is a very artificial sensor, were also working in a more endogenous way. So just to conclude, what I show you today is like the proteome scale induced proximity screens can identify effectors of protein degradation and stabilization in an unbiased manner. The human proteome contains a large cache of effectors and can modulate protein stability. So I mostly talk about E3, E2, and DUBs, but we also looked in the paper at other effectors like the GPI protein, family protein. We also looked at degrams. We identified quite a lot of unexpected hits, so it was very interesting to work on that. And this system can be used to look at degradation and stabilization, but not only. We can look at many biological pathways like transcription, RNA metabolism, protein trafficking, or signal transduction. So I would like to thank the Teipali lab, where I did most of this work, and also our collaborators at Mount Sinai, Anne-Claude Gingras that helped us for the mass spectrometry, and Daniel Schramek for the mouse study, and also my former colleagues that started this project. And I would like to thank you, and I will be happy to take any question.
Thank you, Julian. A huge amount of data generated that will no doubt keep your lab busy for years. We have time for a few questions, so I'll go straight onto that. So first one, great talk. Recently, FBOX2, it was shown as a ligase that can degrade various targets. In your screen, it was shown as a stabilizer. Can you comment on these results? And an additional question, do you think that degradation you can see might be an effective auto-ubiquitination of ligase and degradation of whole complex instead of ubiquitination of GFP fusion protein?
So yeah, the first part of the question, it could be because in our system, degradation is not possible because of the geometry of the system. Also, in our system, the protein is tagged. So everything is C-terminally tagged. So maybe you are impairing the function of FBXO2 in this case. I tried to to move the tag, to switch it for many of the E3, and most of them were unaffected, but some were drastically. So it could be one explanation. The linker size too is another one. So yeah, it's completely possible that in another system, FBXO2 could be used as a degrader, and also the nature of the substrate too. And the second part was ubiquitination of the full complex.
Auto-ubiquitination, yeah.
Yeah, auto-ubiquitination, I cannot exclude that. It would be very cool to look at it. Yeah.
No, thank you. So another question, the existence of BCR-able PROTACs degrading via cereblon seems inconsistent with the synthetic screen. How predictive would you say your findings are?
So cereblon in my hand was working really well as a degrader, but only for specific targets. And many publications are also showing that a lot of targets are resistant to cereblon. So this is not very inconsistent. It was never shown that cereblon can degrade BCR-ABL. And in my hand, it never did, to be honest. So that's my comment about that. And how predictable it is. I think some of these three, like FBXL12 and 15, for example, they are not sensitive to tag location, to linker size, to nature of the substrate. So this one, for example, I can with a lot of confidence predict that they will mostly degrade everything. They don't care, they are not sensitive. But VHL, for example, is more tricky. You need the proper configuration, the geometry needs to work, and the substrate is important too.
Yeah, but thank you very much. So I guess once again, thank you, Julian. And we'll keep the story moving on to our next speaker. So next on the agenda, Charlotte Crow will present on mechanisms of degrader-targeted ubiquitinability. Charlotte joined Alessio Ciulli's lab at the University of Dundee in 2020 as a PhD student. She's originally from Strasbourg and moved to Scotland to study an NChem in chemistry at the University of St Andrews. Her current work within the Centre for Targeted Protein Degradation focuses on using chemical and structural biology approaches to understand the mechanism of small molecule degraders in TPD. Over to you, Charlotte, the floor's yours.
Thank you so much for the introduction, Joel, and yeah, thank you also, Julian, for the talk previously. That was really nice, and it's going to be a tough act to follow. So as Joel mentioned, I'm a PhD student at the University of Dundee. I'm actually only 3 weeks away from my PhD thesis submission, so this is a really timely opportunity for me to present some of the PhD work that I've been doing. And so at the University of Dundee, we're located on the east coast of Scotland, where we have our main School of Life Sciences, which is where I started studying back in 2020. But then in January 2023, we packed up all of our stuff and moved our equipment and ourselves across the road to a new building, which we established called the Centre for Targeted Protein Degradation, also known as the CTPD. So this building is founded and directed by Alessio Ciulli, who's also my PhD supervisor. And once we'd finished celebrating the opening of this new building, and as you can see, Alessio's very apt at opening the sparkling wines for us to celebrate, we were able to move on to our main mission, which is to develop small molecule degraders which hijack the ubiquitin proteasome system to degrade disease-causing proteins. And also importantly, and this is going to be one of the focuses of my talk, to understand the underlying mechanisms in order to develop better degraders. So I'm sure this pathway does not require much introduction for this more TPD audience, but in brief, we all know that the PROTAC must first enter the cell and then engage with its binary binding partners. So it needs to form a binary complex with the substrate and also the E3 ligase, which it's aiming to recruit, before then forming the key ternary complex between the substrate, the PROTAC, and the E3 ligase. The E3 ligase can then recruit the E2 conjugating enzyme before monoubiquitinating and then polyubiquitinating the target, before then inducing degradation of the target by the 26S proteasome. And so for the purpose of this talk, I'm going to focus on the right-hand side of this mechanism and focusing in first on this ternary complex in the center here. And so there's a number of ways that we can study this ternary complex. And the first that I want to focus on is biophysical assays. And so essentially what we're looking to do, and this is what I'm going to present now, is looking at assays to look at ternary complex stability versus target degradation. And so in brief, we've assembled a library of PROTACs which are all based around VHL and the BET bromo domain. So for example, with MZ1, and then we have a number of chemical derivatives based on this. And then two key assays which I'm going to focus in on, and we have built a whole panel of assays to study these degraders, but I'm only going to present two today, are this degradation assay in cells, which was first presented by Kirsten Ruching in 2018, where we do a CRISPR high-bit knock-in of high-bit BRD4 in HEC293 cells. And then when we transfect in large bit and then enduracine treatment, we can get a luminescent construct. And then on degradation by the proteasome, we get a loss of luminescence. So I've just shown an example of the degradation, which is time dependent and PROTAC dependent here with MZ1. And then the ternary complex assay that we've decided to use is with the Helix biosensor. So in this Helix biosensor, we have a biochip which has a gold electrode surface. And so if you visualize here the biochip surface, essentially we have a Y structure of complementary DNA strands. And on these DNA strands, we immobilize a donor dye, an acceptor dye, and then we can also immobilize two proteins. So here we immobilize the BET bromo domain and also VHL. And then when the PROTAC, visualized in yellow here, comes onto the DNA biochip, we get closing of these Y structure arms, and then we can monitor this fret signal which forms. And so just as a brief example here, I'm just showing MZ1 and a few other derivative PROTACs versus cis MZ1, the negative control. And you can see here we get nice fret signal for these compounds. And so focusing on these two assays and with all of our PROTACs, we performed some screening and collected ternary and degradation data. And I just want to focus in on a couple of points which we saw, which really highlight some rules for governing degradability of targets. So these two examples include these MZP PROTACs, which have an IBET726 BET ligand, versus the MZ PROTACs, which have a JQ1 BET ligand. And as you can see from these two plots here, where we look at degradation rate versus PROTAC concentration, we have a much faster rate of degradation with the MZ PROTACs than we do with the MZP PROTACs. And when we plot our ternary complex on rate versus off rate, you can see that all of the MZ PROTACs seem to have a lower KD value, so an increased stability, whereas all of the MZP PROTACs have a decreased KD. And so based on this, we can deduce that actually these MZP PROTACs are mediating less degradation, and that's likely owing to the fact that these PROTACs are non-cooperative. And so the equilibrium will lie towards the binary binding rather than the ternary complex. And by contrast, the MZ1 PROTACs, which are positively cooperative, will lead the equilibrium towards the ternary complex. So that's just one example. Another example that I'm going to show today is the MZ PROTACs versus a number of chemical derivatives that we've made on the VHL ligand highlighted in green here. And so what we did with these compounds was that we essentially generated through structure-based drug design some VHL ligands which would increase binary binding with VHL, and also some which would decrease the binary binding with VHL. So with our helix Y structure, we also measured binary binding with VHL, and you can see here our benchmark MZ1, and then here are the PROTACs where we increased binary binding, and here where we decrease binary binding. And as you can see, when we look at the degradation rates again in pink in the middle, we have our MZ1 benchmark, and then our increased binary binding mediate much faster rates, whereas our decreased binary binding have slower rates of degradation. So these are just two examples out of a lot of data which highlight ternary complex formation and target degradation and how one governs the other, but we do also know that there are some other factors involved. And so these are just a few studies which have highlighted how ternary complex stability correlates with increased target degradation rates and amount. Another method that we can use to study ternary complexes is structural studies, and this is what I'm going to focus on in the next part of my talk. And so over the past few years, there's been a large number of PROTAC ternary complexes which have been solved in complex with the substrate, the PROTAC, and the E3 ligase substrate receptor and adapter proteins. So these are just a few examples which have been solved by crystallography and cryo-electron microscopy. However, these specific studies lack the full Cullen ring E3 ligase assembly, and therefore, based on these, it's difficult for us to obtain any information about how ubiquitination is occurring. So in order to address this question, what we wanted to do was to obtain a cryo-EM structure of the full PROTAC-induced ternary complex with the full Cullen ring E3 ligase. And so as a first step towards this, we did in vitro expression and purification of the full-length E3 ligase, which we also preparatively nedylated and purified. We then incubated this with MZ1, our substrate, and our E2 ubiquitin stable conjugate. And after incubating all of this in vitro by mass photometry, we observed a shift in peak, which corresponds to a shift in mass. So we had full ternary complex formation. We then went ahead and solved our first data set. So we did this in-house at the University of Dundee on our Glacios 200 kV with Falcon 4i. And as you can see here, we now have a lovely structure of the full ternary complex. And what we're really observing here is that the PROTAC is directing the substrate protein towards the C terminus of the E3, which is where catalysis, ubiquitination will occur. We then went on to solve another structure where we then had the resolution to resolve NAD8 here, and then in red the E2, and also with ubiquitin, and we did this in Oxford on the Cryos 300 kV. And what was really exciting about this data set was that we actually had the resolution that we needed to start capturing some of the conformational heterogeneity. So some of you might know that one of the benefits of cryo-electron microscopy is that some of the algorithms that have been published over the past 1 or 2 years, which are very new, exciting, and recent, mean that we're now able to capture the conformational landscape of some of the proteins that we're solving structures of. So while this that I'm showing here is a consensus volume, really this structure is made up of a number of different poses which are present in the system. And so you can see with these two videos some of this conformational heterogeneity, where we can see the culin ring E3 ligase, which is hinging in two different axes, which was really exciting for us to see. We were then interested to look at the interface between the substrate and the E2. And what we observed was that there were 3 lysines, highlighted in pink here, which were in very close proximity to the active site cysteine for ubiquitination to occur. And so in order to understand the importance of these 3 lysines, we decided to do an in vitro ubiquitination assay. And then my colleague Sarah Chandler, who is a PhD student in Ron Hayes' lab, was able to excise the bands and do mass spectrometry of the ubiquitination sites in this in vitro assay. We were able to find a number of lysines which got modified, but when we mapped these onto the cryo-EM structures that we just solved, what we noticed was that all of the lysines highlighted in pink here are actually showing evidence of modification in the in vitro ubiquitination assay. And in contrast, when we flip the structure 180 degrees, we can see it now in blue, all of these lysines showed no evidence of modification in this in vitro system. So we were essentially able to draw a line in the substrate between what we call the light face, so where this ubiquitination is occurring and where access to the E2 is possible, and this dark face where we did not find any evidence of modification by mass spectrometry. And then for anyone who has studied in the ubiquitin field, as you'll know, one of the classical things to do is to do lysine to arginine mutants. So we did an 8-point lysine to arginine mutation on all of these lysines highlighted in pink. And then we also noticed from our in vitro ubiquitination and mass spectrometry that this K456, which appeared to be optimally positioned to access the active site cysteine, was the only lysine that was modified after 30 minutes. And it was actually the one that showed the highest intensity of modification after 3 hours of in vitro ubiquitination reaction. So we also did a single point mutant for this single lysine, and then we also did a triple point mutant with all 3 of these lysines, which seem to be close and closest to the active site. And so we again turned to our in vitro ubiquitination assay. So we expressed and purified all of these mutants recombinantly, and then we labeled them fluorescently with an Alexa fluor dye. And as you can see, the wild type has nice ubiquitination, whereas the 8-point mutant has much reduced ubiquitination. We were then able to quantify this, so monitoring the conversion of unmodified to modified substrate. And in green here, you can notice that the wild type substrate has a very good ubiquitination. However, in contrast, the triple point and the 8-point mutants had much significantly reduced ubiquitination. So we thought, okay, clearly there is some relevance from our in vitro mass spec and this ubiquitination reaction, but we then wanted to look at what the relevance of these lysines was in cells. So my colleague Connor Cragen, who's also in Alessio Chili's lab as a postdoc, was able to set up this cellular degradation assay. So again, using these exact same substrate mutants, but now with a nanoluke tag. And he transfected these into HEC293 cells and then treated them with MZ1 for 4 hours and was able to get this degradation profile for all of these substrate mutants. And so you can see in green at the bottom here, we get quite good degradation in cells. And then, for example, in pink here, we get quite significantly reduced degradation. And so clearly there was some kind of difference, and we were also able to quantify DC50s and Dmax values for this and show that they were indeed statistically significant. But nevertheless, we were still perplexed as to why this triple point mutant was still getting degraded so much. And so one of the rationales for that could be the fact that this nanoluke tag does also have some surface lysines, which could get modified, inducing degradation of the whole construct. But to just go a step further and really understand ubiquitination in a cellular context, my colleague Gajanan Saif, who started as a postdoctoral researcher in Alessio's lab several months ago, was able to set up cellular ubiquitinomics. And so he treated some cells with MZ1 for 15 minutes and was able to analyze the KGG modified peptides by mass spectrometry. And what Gajanan saw was that all of the lysines in pink here actually map to the exact same lysines that we'd originally identified as being ubiquitinated in vitro. However, he also identified an extra 2 lysines which we'd previously assigned to our dark face, which may be also accounting for some of the cellular degradation that's occurring. And so with these two lysines, one of these in particular is really located on the cusp of the light versus dark face and pointing in towards the light face. So we could have assigned this to the dark face originally. However, K431 is actually located quite further into the dark face. And so I think what these modified lysines in cells indicate is that actually our system, which is very flexible with this inherently flexible culling ring E3 ligase, are able to access lysines which are potentially suboptimal. And so ultimately, what is all this for? Ultimately, we want to understand what makes a good degrader. And so one thing that we do know is that ternary complex formation between the substrate, the PROTAC, the E3 ligase is a requirement. And what our study here has shown is that MZ1 directs the substrate towards the E2 catalytic site for ubiquitination. We've also seen in this study that these lysines are spatially optimal to access the E2 catalytic site. However, since MZ1 forms such a stable and long-lived ternary complex with the BET bromodomain BRD4-BD2 and VHL, the fact that the residency here is so long, coupled with the fact that the culling ring E3 ligase, as we showed by cryo-EM, is inherently flexible, means that suboptimal lysines can also get captured and ubiquitinated in this system. And so how is this going to inform us on how to design PROTAC degraders? Well, one thing to start thinking about is how PROTACs engage their substrate and direct them towards the E2 for ubiquitination. And can we already determine any rules based on this? Well, we could hypothesize that if our degrader engaged the target and pointed it away from the active site cysteine in the opposing orientation, then this could get less ubiquitination. And so more studies with more PROTACs will help us to really see if this hypothesis is viable and understand exactly what the requirements are for PROTAC-mediated substrate orientation. And so with that, I'd really like to thank my supervisors, so Alessio Trulli, Ron Hay at the University of Dundee, and Hannah Maple at Bio-Techne. I'd also like to thank some of the lab members who have contributed to this study. So in particular, Mark, Sarah, Connor, and Gajanan. And lastly, I'd like to give a shout out to some of the infrastructure access, training, courses, and networks that I've had access to during my time in Scotland and in Dundee. Cryo-electron microscopy over the past couple of years in Dundee has really started to become a booming method. And so I've been extremely lucky to be able to start riding that wave and benefit from all of the training and courses that have been available to me. And finally, my funding, the MRC and Bio-Techne. And thank you all for your attention.
Great job, Charlotte. That's fantastic. I'm sure the audience has some questions, so let's take a look. Yeah, very nice talk. Do you think that the difference between PEG and alkyl linker MZP versus MZ protax might be caused by hydrophobicity of alkyl linkers? They prefer to be shielded from water, and the driving of the ternary complex for stability is more driven by hydrophobic effects than PPI interactions between the proteins.
Yeah, so the MZP protax also have a PEG linker actually. So they're exactly the same series of protax as MZ, except that they differ in their substrate ligand. So we just have a IBET726 ligand instead of a JQ1 ligand. Okay,
so thank you. Do you have any suggestions based on your structural work to provide a cutoff distance between target protein lysine and the E2 catalytic site beyond which ubiquitination is impaired or doesn't happen?
Yeah, well, that's exactly what we're going towards. So this is just the beginning of these, structural full-length ternary complexes where we're finally able to see in a more reliable way what the interface looks like. But as soon as we start testing more protax, which we're looking to do and is ongoing, we should be able to see how all of these orient their substrate, and then coupled with this mass spectrometry methodology, then we'll be able to really start to measure these distances and map out the actual requirements.
Fantastic. Yeah, so more work to come by the sounds of it. This is just the beginning, but it's fantastic. So I guess once again, thank you for the great talk and Q&A, Charlotte. So we're out of time for this talk, but yeah, we'll keep the show moving on. So this is on to our next session, which will be presented by Professor Tom Smithgull. So Tom is the William McElroy Professor of Biochemistry in the Department of Microbiology and Molecular Genetics at the University of Pittsburgh School of Medicine. He earned his PhD in pharmacology from the University of Pennsylvania School of Medicine, following postdoctoral training at the National Cancer Institute, NIH, where he held faculty positions at Georgetown University and the University of Nebraska Medical Center before joining the faculty of the University of Pittsburgh in 1998. His current research interests center on protein tyrosine kinase signaling and drug discovery related to HIV, AIDS, and myeloid leukemias. He has authored or co-authored more than 150 peer-reviewed publications to date and has been continuously supported by NIH grants since 1993. He's also inventor or co-inventor on 6 patents related to HIV-1 NEF inhibitor development as a new approach to AIDS therapy, including a recent patent filing on the NEF protax that are the subject of this talk today. And with that, Tom, take it away.
Great, thank you. I appreciate the invitation to speak today. And I'll shift the focus a little bit to infectious disease targets, which hopefully you'll see represent, in a sense, ideal targets for PROTAC development. So without any further ado, sorry, just trying to get the slide to advance. Here we go. Okay. So just as a quick reminder about the status of HIV and AIDS, we're now more than 40 years out from the first reports of HIV-infected individuals. And it's the global pandemic continues to persist worldwide, as you can see from the data, close to 40 million infected individuals, more than 1 million new infections per year, and unfortunately over 600,000 related deaths. These are primarily in Africa and other developing areas. Now, so I'm sure everyone is aware, there are many, many FDA-approved drugs to suppress HIV viremia, and these, of course, have turned the infection from a death sentence into a chronic manageable condition. However, none of the existing therapies eradicate so-called viral reservoirs, and as with any chronic therapy, this can lead to drug resistance plus its own comorbidities. So what we really need are new drugs and new approaches to clear the virus and essentially functionally cure the disease. So the focus of this talk today is a viral accessory factor. It's called NEF. Just to back up, existing antiretroviral drugs target familiar HIV enzymes, reverse transcriptase, protease, integrase, and so on. NEF, on the other hand, is an accessory factor, and as you'll see, it's not an enzyme, and that makes it somewhat of a difficult drug target. But nevertheless, it has an important role in HIV replication and disease progression, and as you can see on the slide, it promotes infectivity, the replication cycle and spread of the virus. It can also, importantly, promote immune evasion of HIV-infected cells, in part by downregulation of MHC1 and antigen presentation. It also protects so-called latent reservoir cells by upregulating the immune checkpoint receptor PD1, and in both animal studies and in elite individuals that have been infected long-term with the virus but don't progress to AIDS, we know that NEF is sometimes missing, suggesting it has an important role in progression. So all these factors together, sorry, there's kind of a delay here. There it comes. Okay. So the conclusion here is that if we were able to develop inhibitors of NEF, we'd have the potential to not only interfere with the viral replication cycle, but restore host immunity. For example, the cytotoxic T lymphocyte response to help cure the virus, clear the virus. All right. So a little bit more about NEF in terms of its structure. NEF is myristylated and associated with plasma membranes, and that's essential for its function. Myristylation occurs on this N-terminal anchor domain and is aided by a basic patch along the anchor. This then leads into this folded core region, so this purple domain, and then NEF has multiple motifs or surfaces that are involved with interactions with host cell proteins that account for its function. So many NEF interactors have been reported in the literature, and I'll just mention a couple briefly that are important to this talk. So the way we got into this field was many years ago, we and others discovered that NEF likes to interact with non-receptor tyrosine kinases, first of the SARC family and then later the TEC family, and that these interactions lead to constitutive kinase activation, which helps viral transcription and replication. And this is important to highlight. NEF in particular interacts with a SARC family member called HCK, and this interaction is something we studied in detail and ultimately harnessed as an assay to identify small molecule binders of NEF, which were the basis then for PROTAC development. So I'll get into that in just a few minutes. The other key class of proteins that NEF interacts with are the tails of several receptors, including MHC1, which I already mentioned, as well as viral receptors, CD4, X4, R5, as well as the restriction factor, serine5. And in addition, or what it does is it bridges these receptors to the adaptins that are involved in endocytic trafficking and receptor downregulation. And this is the molecular basis for immune escape, as well as suppression or reversal of restriction. Okay, so a key point here is, as already mentioned, NEF is not an enzyme. It doesn't have an active site, and you can see from the illustration, it uses different surfaces to interact with host cell effectors for function. So how do you come about finding an inhibitor? Or as we'll see, the better strategy is just to degrade the protein, get rid of it completely. Okay, so how do we find small molecules to bind NEF? And to do this, we took advantage of what we knew about how NEF can activate a kinase. And that kinase is called hematopoietic cell kinase, or HCK. It's expressed in macrophages, which are an HIV target cell. And this cartoon illustrates what we know about the activation mechanism. So NEF, as I mentioned, is myristoylated. It inserts in the membrane and it actually forms homodimers. And then it can recruit HCK through its SH3 domain. And if you're not familiar with the Sarc family, these are multi-domain structures. They have a typical bilobed kinase domain, and then they have two regulatory domains, Sarc homology 3 and SH2, and these two in tandem help keep the kinase in the downregulated or off state. But what happens when NEF binds is it displaces the kinase domain from the SH3 domain, and then because it's a dimer, it can also form this complex to promote kinase activation. And in terms of virally infected cells, that helps or enhances viral transcription. So Lori Sedlak in our lab a number of years ago was able to, in a sense, reduce this to a very straightforward in vitro assay system where we made recombinant HCK in the downregulated state and then combined it with purified NEF in the test tube. And what you can see in this slide is that as you add NEF to the assay, you get increased activity. I'm sorry, as you add HCK to the assay, the kinase. But when you do it in the presence of NEF, you can find a point where activation of the kinase is dependent on NEF and not the kinase alone. And so this, in a sense, allowed us to run high-throughput screening assays looking for small molecules that interfere with HCK in the presence of NEF. So in a sense, we're using the kinase as a reporter, and kinase assays, of course, are very amenable to HTS. So this is all published. I'll just go real quickly here. Working with Paul Johnston in our Drug Discovery Institute here at Pitt, we were able to screen a medium-sized library, about 200,000 compounds. We got 1,300 raw hits, and then we rescreened these in concentration response to find the ones that were dependent on the presence of NEF, because we didn't really want kinase inhibitors. We wanted small molecules that bound directly to NEF to interfere with the kinase. And at the end of this, we found 62 compounds that met those criteria. Excuse me. And then Lori screened all of them for inhibition of NEF-dependent activation of HIV replication in cells, and found a number of compounds. The most important, though, is this one boxed here called B9, and you can see it's a hydroxypyrazole with these two phenyl rings attached. And this compound, I don't have all the data in here, but it binds directly to NEF. It interferes with NEF-dependent enhancement of HIV replication, and also it shuts off NEF-dependent activation of kinases. And so moving forward at that time, we then collaborated with Mario Ostrowski at the University of Toronto, and using that original hit compound and then two non-diazo derivatives, we were able to show that, yes, indeed, if you add the NEF binder, you can partially restore MHC1 to the cell surface. And then when you add autologous CD8s those actually recognize and then kill the virally infected cells. So this was a very important proof of concept that inhibiting NEF may provide a way to restore immune function and help clear the virus. Okay, so from there we wanted to make better compounds, and here we teamed up with the Fox Chase Chemical Diversity Center in Doylestown, Pennsylvania, and the lead chemist on this was Colin Tice. And he took that original B9 hit compound, and then if you look at the structure, there basically were three arms, green, red, and blue. And from that, he made a whole series of analogs, more than 200 analogs. And then we screened these for NEF binding using surface plasmon resonance and also for antiretroviral activity. And from this, we found a number of analogs that bind tightly, nanomolar to picomolar KD values, and showed that they inhibit HIV replication in the nanomolar range. And these are just a couple examples, and you can read for yourself. They're IC50s against the virus. They do this with a fairly good therapeutic index, and then the KD values are listed below that. But the key point here is that none of these analogs, although they're good antiretrovirals, they weren't particularly good at rescuing cell surface MHC1, and they had even weaker impacts in terms of rescuing other receptors, including CD4. And really what we want is something that can shut down all NEF functions, and that's what led us to the idea of a PROTAC. So this is the slide I think we'll see in every talk today. So essentially what we wanted to do was use our existing NEF binding compounds and then couple them to E3 ligase recruiters, and then hopefully trigger ubiquitination and targeted degradation of NEF. And there are two advantages. Well, one additional advantage to this. One is, of course, if we eliminate NEF, that will get rid of its functions. And it turns out that NEF is also, or NEF-derived peptides are good antigens for the CTL response, and so the proteasome may help with that as well. Okay, and then this just sort of summarizes what I just said in one slide. So when NEF is present, all these receptors get downregulated, kinases get switched on, PD-1 is induced. But if we degrade NEF with a PROTAC, we can restore receptors and restriction factors and also switch off the kinase. Okay, so based on that premise, Colin synthesized a library of about 100 analogs, about 2/3 based on cereblon targeters, so thalidomide analogs, and the remainder to VHL. Not shown here, but we did some preliminary work where we added linkers at various positions along the NEF binder and then ran SPR to make sure they didn't interfere with NEF binding. And not shown, but the best positions for attachment were here and here on these two phenyl rings. And then, as is typical, we used a whole series of linkers of different chemistries and lengths and so forth. Okay, so the next job was to screen for ubiquitination of the NEF protein. So here we used this ubiquitin nanobread assay from Promega, tagged NEF with nanoluc, and then ubiquitin is provided as a halo-tagged molecule. So in transfected 293 T cells, when the PROTAC is present, you get proximity effect, and then when you add luciferin, NLuc then is in a position to stimulate the halo tag, and you can measure both the emission from halo as well as the NLuc signal, and then express that as a ratio. And then here's some examples of the data just to show that this in fact does give a readout for ubiquitination that's dependent on ubiquitin. One thing to notice too is in the absence of a PROTAC, there's a fairly strong basal signal suggesting that cells do in fact target NEF for ubiquitination in the first place. And this has been reported in the literature, although the E3 involved is not entirely clear. Also, we only see this increase in signal when halo is combined with ubiquitin. And then in the presence of active PROTACs, you can see there's a significant increase in ubiquitin incorporation. So we screened all the assays, we meaning Lori, and here's just a plot showing, here's the breakdown of the library that I mentioned. And what we found is that all of the actives, in fact, were coupled to CRBN binders, and none of the VHL ones were active, at least in this screen. So the next step was to confirm this to show that the molecules actually do in fact generate or induce NEF degradation in a relevant cell context. So for this, we used a CD4 T cell line called CEMT4, and Sherry Hsu in our lab, she engineered these cells to express GFP tag NEF under the control of a DOX-inducible promoter. And so what you're looking at here is the proportion of cells that express NEF GFP by flow, and then as a functional readout, we could look at downregulation of cell surface CD4, which is, as I mentioned, a well-characterized function of the NEF protein. So as you increase the concentration of DOX, you get more and more NEF expressed, and as a consequence, CD4 goes down. So using this system, we were able to do a couple of things. First of all, just straightforward Western blots to look at activity of, these are the individual PROTACs from the previous slide that induce ubiquitination of NEF in the nanobread assay. And you can see that at least at 3 micromolar after, I believe, 48 hours, there's near complete degradation of NEF GFP in almost every case. So here's in the absence of DOX, induced with DOX, no PROTAC, you get a strong band. And then when the PROTACs are around, the protein is degraded. Okay, then what about phenotypic effects? So we looked at both CD4 and MHC1 downregulation in the cell line. And then here's just one example of a flow result. So this is a 2-dimensional flow diagram where we're correlating levels of CD4 with expression of NEF GFP. And so this is the starting point. If we add DOX, we induce NEF. GFP signal goes up and the CD4 levels start to go down. If we do it with an active analog, it just looks like the control. And that's the same, one of the same analogs that induce degradation by Western blot. And then here's a related but inactive compound, and you can see it has no effect. And then this is just a bar chart showing that most of the compounds, but not all, identified in the nanobread screen, in fact, were able to rescue not only MHC1, but we're very good at rescuing CD4 as well. And as I mentioned, that's not something that we saw with just the binder alone. And then here's, among all of these, the most active compound, 14369, with a DD50 of about 160 nanomolar, and it does induces degradation without cytotoxicity. Okay, so what about mechanism? Well, our finding, and I think this is true for many in the field, that there wasn't a clear SAR with respect to length of the linker, chemistry, and so on. But one thing we did notice is that our active PROTACs, when they bound to, this is just NEF, it's not ternary complex like we saw in some earlier talks, but we can see there's a sort of a steady on, and then when we switch to wash, sorry, this is by SPR, that the compounds remain stably bound, at least over the time course of the SPR run. On the other hand, here's a very similar compound that's inactive. You can see the only difference chemically is this linking oxygen here, and here it's just a carbon atom. And this molecule binds very rapidly, but then dissociates very rapidly. And so this may suggest that residence time on NEF is an important predictor for PROTAC action. And there are more examples of this in the publication. Also, I should point out that all the SPR, we did SPR in all of the analogs, and that was all done by Haibin Shi, who's our resident SPR expert. And then another point of mechanism was to ask, well, can we demonstrate ternary complex formation? And so in these initial studies, John Jeff Alvarado in our lab expressed and purified the thalidomide binding domain of CRBN and basically did some analytical SEC experiments. So if we run NEF by itself or CRBN by itself, or the two together, you can see they all co-elute. So the molecular weights, or the retention volumes anyway, are roughly the same. But what's interesting is when we pre-incubate NEF with CRBN plus one of the active PROTACs, we get a new peak of a smaller retention volume, meaning that it's inducing formation of a higher molecular weight complex. So this is exciting, and it's kind of a prelude to X-ray crystallography to try to get at the structure of the active complex. And then finally, I want to mention that these PROTACs do indeed have, they maintain the antiretroviral activity that we observed with the NEF binders alone. So we did these experiments in two ways. One is using a reporter cell line called TZMBL. So these are HeLa cells where the HIV LTR, in a sense, its promoter is linked to luciferase. This is very commonly used in the HIV field if you're not familiar. And here we're titrating in either active PROTAC in red or an inactive analog in gray. And you can see that the active compound blocks infectivity in a similar manner or at a similar concentration as we observed for its DD50. And then perhaps more importantly, if we take primary cells from normal donors, peripheral blood mononuclear cells, and then infect them with HIV-1 in the presence or absence of NEF, PROTACs, in every case except for one, we can knock down replication to the levels that we see with a NEF defective form of HIV. So this is good evidence that but yet to be shown that they're working in a relevant context, at least in an in vitro HIV replication assay. Okay, so here's where we're going with this. I think a key point for us is to move ahead and see whether degradation of NEF not only has antiviral activity, but can it actually reestablish immune responses through the mechanisms that I mentioned. And to do this, we need to develop analogs with good metabolic stability and PK, and then the plan is to test them initially in a humanized mouse model of HIV replication and latency. I didn't show the data, but we've also tested some of our PROTACs against SIV NEF, and they seem to show activity as well. And so we're hopeful we may be able to identify an analog that would be useful for SIV-infected non-human primates. We need to map the lysine or lysines that are involved in attachment of ubiquitin, and the approach is very similar to what we heard in the previous seminar, with a combination of mutants and also proteomics. And this is important, and the hope would be that perhaps multiple lysines are involved rather than a single lysine, because that single lysine may be more susceptible to escape mutants. And then I already mentioned that we're working hard on getting a ternary complex of NEF with the PROTAC, and also we're actually using now the MIDI CRBN construct that was recently reported. And then finally, we're also assessing overall on-target selectivity using standard proteomic approaches. So let me just finish then by just pointing out everybody that's really driving this project forward. So I mentioned Lori. She's done all the screening assay development, both for the initial binders and subsequent ranking of the PROTAC analogs. John Jeff, who's made recombinant proteins and is working on the crystallography. I've done all the SPR and developed those assays. Also Sherry and Lee, who worked on the inducible cell line, as well as animal models. And then finally, just to shout out to Fox Chase. These are our medicinal chemistry partners, and they're responsible for synthesis of all of the initial binding library and then the PROTACs. And this work has been supported by NIH, NIAID, and the STTR program, at least in part. So I hope I didn't go over. Thank you.
Thanks, Tom, for a really enlightening presentation. No, we have time for a few questions. Yeah, do you have any hints of benefits of inhibition versus degradation for this or other antiviral targets, and then thoughts on where the benefits might lie?
Yeah, I think in the paper, and I didn't show this slide, but we did a head-to-head of binders versus PROTACs, looking at downregulation of MHC1 and also downregulation of CD4. And what we found is, well, a couple things. First of all, the amount of NEF protein in the system is a big determinant of the outcome. And that's where the inducible system is very important. Because if you overexpress too much NEF, it's very difficult for either drug format to antagonize it. But having said that, when you compare a binder occupancy-based inhibitor versus a PROTAC, at sort of an appropriate level of NEF expression, MHC1 is similar, CD4 not so much, but the PROTAC is very effective against both. And I didn't have time to get into all this, but the AP1 and AP2 pathways are actually responsible for not only CD4 and MHC1 downregulation, but many other receptors that are important to NEF activity. So the promise is that targeted degradation may reverse all of these functions of NEF and hopefully boost effectiveness, say, of a vaccine or monoclonal antibodies.
Yeah, thanks. I'll squeeze one more in, actually. So great talk. Do you know the half-life of protein resynthesis for NEF?
Yeah, great question. And that's something we're working on that. It's preliminary experiments suggest that, at least in transfection systems, which may not recapitulate what's happening with the virus, but just for the sake of answering the question, the levels are maintained pretty highly. And it's just that extra tip, perhaps, with the PROTAC that switches that balance more to a degradation in relation to synthesis. But that's a great question and something we want to explore more carefully, especially in the context of HIV-infected cells.
No, thank you. There's a few more questions that have come in, so we'll assign those across to you and you can maybe answer them via the chat function. But thank you once again, Tom. That was a fantastic talk. So that's all we have time for in the first half of the symposium. So we'll now have a short break where we'll be back at, I'm in the UK, so it's 4:31, so we'll be back at 4:35, so 4 or 5 minutes. So welcome back. It's time for me to welcome our next speaker. So Brianna Zerfas is currently the biochemistry and structural biology group leader and senior scientist at the Center for Protein Degradation in the Dana-Farber Cancer Institute. She received her PhD in chemical biology from Boston College in 2017, working with Professor Jianmin Gao on the design and synthesis of peptide antibiotics with unnatural amino acids. From there, Brianna moved to the Department of Medicinal Chemistry and Molecular Pharmacology at Purdue University as a postdoc with Darcy Schrader. While at Purdue, her research focused on the screening and development of fluorescence-based probes selective for various isoforms of the proteasome to be used in live cells. In April 2020, Brianna returned to Boston to work as a scientist at the Center for Protein Degradation, focused on the new assay and chemical biology tool development before being promoted to group leader in the summer of 2022. Brianna is a member of the organising committees for the Dana-Farber Cancer Institute TPD webinar series and the Women in TPD and Induced Proximity group. So before we hand it over to Brianna, just a quick reminder that you can submit questions for our speakers by using the question box on the right-hand side of the screen, and we'll take questions for each speaker following their presentation. So with that, let's pass it over to Brianna. Over to you.
Great, thanks Joel for the introduction and for the invitation to speak today. So I'm excited to present some of the recent work that we've been doing at the Center for Protein Degradation around a new tag that we've been using for our degrader development, which we refer to as the HiBit SPY tag. So as I think most of the speakers have also mentioned, this is likely not an introduction that this audience will need, but I'm just going to kind of go through what we look at for protein degradation, and specifically the differences as we see it between molecular glues and PROTACs. So for targeted protein degradation, these two different types of compounds have the same end result, but they behave very differently in terms of how you design them and their function overall in the protein-protein interaction. So both systems will include an E3 ligase and a protein of interest. Typically with a molecular glue, you have a much more pronounced protein-protein interaction at the interface, as this glue is small enough to bring the two proteins closer together, whereas with a PROTAC, you typically have a heterobifunctional, which contains an E3 binding portion and a protein binding portion joined together by a short linker. Both of these eventually result in the ubiquination of the protein target, which can then result in its proteasomal degradation and the recycling of these compounds to be able to act as a catalyst for degradation throughout the cell. In addition to some of the pharmaceutical benefits that we've seen in earlier talks today, degraders are also showing a big benefit as a tool compound, because it allows for the rapid chemical knockdown of different proteins, which you can then recover upon the removal of the compound or the washout. And this is done through various different tags, including things like DTAG and Halo tag that have specific PROTACs against those tags. So another technology that I think most people in the audience should be familiar with is the HiBit technology developed by Promega. So this is basically a short 11 amino acid tag that you can CRISPR into your protein of interest. And it's a separated enzyme system where you have your HiBit and then an 18 kilodalton large bit that upon recombination produces an active luciferase enzyme. And as you can see, this has a very tight KDE. So essentially, as long as they're close enough together, they will recombine to make the active enzyme. So Promega has developed this as a technology, including CRISPR-Cas9, where you can add HiBit into the endogenous region of your protein of interest, so that you can use a system including large bit and a luciferase substrate in order to get a readout on the endogenous levels of your protein. So this offers a way to then look directly at your protein in a given cell line, as opposed to needing an overexpression. So here I'm just showing an example of how this is done with BRD4, everybody's kind of favorite degradable protein, where as you have an increased concentration of DBET6, which is a BRD4 specific PROTAC, you see this decrease in signal and eventually bottoming out. And in this range, you're starting to see that typical hook effect, maybe at the top of this range. And this correlates nicely with degradation that you can see through Western blot as well. One of the major advantages of being able to have a system like this is that you can optimize this then to a higher throughput method, including being able to downsize to both 384 or 1536 well plates, so you can do high throughput screening, as well as kind of understanding the degradation around your protein of interest. So this has been really useful, I would say, throughout the field in order to really look at endogenous levels of degradation without needing to do, more cumbersome experiments like a Western blot. But we were trying to see if there were ways that we could take this tag and make it a bit more functional. So have something else added to it so that we would have the high bit readout, but also have a functional portion of the tag as well. So the way that we decided to approach this is by using another split protein system that we're familiar with called SpyCatcher and SpyTag. So in a similar way as high bit and large bit, this protein is split into two pieces, one of which, known as the SpyTag, is a shorter peptide label. When the SpyTag and the remaining portion of the protein, called SpyCatcher, are brought in close proximity, there's a spontaneous covalent reaction which recombines the protein. So essentially anything that carries your SpyTag can be labeled by a corresponding SpyCatcher protein. So we took then these two tags and combined them together. So now this, with the 11 amino acid high bit, we have got a little over 20 amino acid tag, which is really still in a good range for an endogenous CRISPR knock-in. So that's exactly what we did, where we took this high bit SpyTag and knocked it into our HEC293 T cells. And then with the idea that we could transiently transfect a SpyCatcher protein, in this case, we're using a D-tag SpyCatcher, which would then label our protein of interest and then give us this D-tag as a degrader handle. So just kind of to go over, a more visual representation of how this works. So we have our protein of interest. You can label high bit SpyTag on either the N or the C terminus. And this will then get labeled by SpyCatcher D-tag upon transient transfection of that protein plasmid. If you then add a D-tag specific PROTAC to your cells, you can recruit your E3 ligase and have a resulting ubiquitination of your protein of interest, then leading to degradation by the proteasome. By using the high bit, large bit system, you can then get a direct readout of how much protein was able to be degraded using this tagged system, but with a much easier way to knock in your tag compared to D-tag. So as our first kind of proof of concept, we were able to generate a high bit SpyTag BRD4 cell line in HEC293Ts, which we then transfected with D-tag SpyCatcher. After about 24 hours, we then lysed the cells and analyzed by just simple Western as a way to look at how much labeling we were able to see of BRD4. So again, just a representation of the reaction that's happening here. We have our D-tag SpyCatcher, which is about 25 kilodaltons, which can label BRD4. And since SpyCatcher is a covalent label, we should be able to see this, this full complex on a denaturing gel. So we first ran, for example, the non-transfected cell line just to see where our high bit SpyTag BRD4 was traveling on the simple Western system. And what we are able to see is a slight shift in that transfected cell line, where we're seeing then an increase in the molecular weight, suggesting that we are seeing labeling of BRD4 in this case. However, because of the overlap between the peaks, we weren't able to get a full quantitative measure of how much labeling we were seeing in this case. So then to determine whether this labeling did allow us to have degradation of our desired protein, we treated with a few different PROTACs. So as I showed earlier with high bit BRD4, the blue here is corresponding to a non-transfected cell line. We first used DBET6 just to demonstrate that we weren't disrupting anything with our high bit SpyTag in regards to being able to degrade a BRD4 specific with a BRD4 specific PROTAC. And then we tried 2 different DTAG-based PROTACs. So these carry the ligand for DTAG, shown here in red. And just for the sake of looking at various E3s, we use both this cereblon-based PROTAC DTAG13, as well as the VHL-based DTAG VHL. So as we can see, without the transfection of spycatcher DTAG, we don't see any loss of BRD4 in these cells. So we then did this transfection for 24 hours, and we see very little effect on the degradability of BRD4 using DBET6, which is to be expected. But this is just a good indication that we're not interfering with the ability of BRD4 ligands to bind the protein by adding on the DTAG here. And then what we're seeing in the case of both of the DTAG PROTACs is now we're seeing a very good potent degradation of the complex in total. And what we believe is happening here in terms of the Dmax is that this will correspond to the percent labeling of the protein. So we're essentially, I think we're degrading all of the labeled BRD4 in these cases. So as I mentioned, BRD4 is, a really good proof of concept, but we were hoping to use this system as a way to approach proteins that didn't have known degraders that you could use as a positive control in a screening standpoint. So we turn to look at IRE1 alpha. So IRE1 alpha is one of the ER stress recognizing proteins that responds to the unfolding protein response. So in this case, when there's ER stress recognized in the cell, you see an increase in unfolded protein, and IRE1 alpha is one of the systems in order to be able to overcome this. So upon the recognition of these unfolded proteins, you get a dimer of IRE1 alpha forming through the auto transphosphorylation of the 2 monomers, and then this activates the RNase activity of IRE1 alpha. Through the splicing of XBP1, we then see the upregulation of various different proteins that will respond to that will increase the response to the unfolded proteins to allow for the disposal of these unfolded proteins in the ER. So we focused in on the cytoplasmic portion of IRE1 alpha, and that's because it contains 2 different domains. So in blue, I have showing here the kinase domain, which is responsible for the trans autophosphorylation. So the substrate of IRE1 alpha then being a second monomer of IRE1 alpha. And then in red, I'm showing the RNase domain. So this is where we have the activity on the XBP1. So focusing in on these 2 domains, there's been several different links of IRE1 alpha to different types of cancer, which is understandable, as you would imagine. With the increased demand of proteins in cancer, you would then need to compensate for this, but then increase in the response to ER stress if these proteins are being misfolded. So there's been quite a bit of research around different ligands to target IRE1 alpha. On the kinase domain portion, there are IRE1 alpha specific KIRA compounds, and there's also some clinical compounds that have been shown to have inhibitory effects on IRE1 alpha as well, including sunitinib. On the RNase domain side, there's several different options where you're actually using the aldehyde in these different compounds to engage a specific lysine in the RNase domain as a way to inhibit. So these are much smaller and reversible covalent compounds. And here we actually have MKC8866, which is currently in clinical trials for different types of breast cancer. So there's a good representation of this as a viable target within cancer. The reason we were interested in it from a PROTAC standpoint is because there's becoming additional evidence that there's scaffolding functions that occur from IRE1 alpha, as you see dimerization and oligomerization at the ER. So we thought a PROTAC would be a more effective way to be able to target this within cancer. So we first wanted to demonstrate that IRE1 alpha was in fact degradable. So being, a trans ER membrane protein, we weren't sure what was accessible for it in terms of E3s and whether degradation in general would be feasible. So ahead of starting a PROTAC design, we first knocked in our high-bit SPI tag, going through a very similar system as we had done for BRD4. So we can see here by looking at Western, we actually have two different specific clones we were able to collect for this in HEC293Ts. And upon transfection of these clones with DTAG SPI catcher, we can actually resolve those two bands. So what we're seeing here is around 70 to 80% labeling after a 24-hour transfection. So yes, corresponding to the higher molecular weight band. So then in using DTAG 13 as a PROTAC treatment here, we can see a very nice dose response that against both clones after transfection. And as I mentioned earlier, the Dmax then corresponds well to the level of protein labeling that we're able to see. So that was, again, demonstrating that any protein that's labeled, we're able to degrade within this system. Okay, so then we moved to develop a PROTAC within the system. So we started by modeling a ternary complex between IRE1 alpha and cereblon. We were primarily focused on the RNase domain, as that was a bit further away from the membrane, so we expected it to be a bit more accessible for E3 ligases. And we used just a simple salicylaldehyde inhibitor, one of the ones I had shown on the previous slide, as then this would give us a much smaller starting point so that our molecular weight of the PROTAC wouldn't grow too quickly. So from the ternary complex modeling we did, we found that they could be, the PROTAC length would be suitable to be very short. So we were actually getting some favorable protein-protein interactions between cereblon and IRE1 alpha, suggesting we could have a much shorter linker without causing any sort of clash. So we included that in our PROTAC design and came up with this compound we named CPD2828. So here I'm showing the cereblon ligand here, again, straight away from the traditional IMiD, as we were looking to have a shorter distance, just a really small linker here. And then this is the salicylaldehyde inhibitor for the RNase domain. In addition to our PROTAC degrader, we also made the inactive and methylated PROTAC as a way to demonstrate that the effects we were seeing were due to engagement of cereblon and not just because of destabilizing the RNase domain. So we look at these two PROTACs in our cereblon engagement assay. So this is something we've developed in-house as a cereblon nanobret, where we can look at the displacement of a fluorescent ligand to look at cellular-based engagement of cereblon. So we use CC220 here as a positive control, and we're getting pretty good engagement with 2828. And as I mentioned, we weren't expecting our negative control 3121 to engage cereblon here at all. So that was promising to see that in cells we're not having any effect of binding to cereblon. So then when we move to degradation at 24 hours, we can again see the that we're getting specific degradation just with 2828, and that our negative control is not having any impact on the levels of IRE1 alpha in this case. And in addition to HiBit, we did also look at this by the Just Simple Western as well, just to show that this was similar to what we are seeing in the engineered cell line. So this is just in the parental hex cell. And we actually see a bit more degradation Dmax with the parental cell line compared to our HiBit system. In addition to that, we wanted to look at the selectivity across the proteome of our PROTAC. So we treated HEC293 T cells with 10 micromolar of our PROTAC, and what we're seeing here is pretty selective degradation of IRE1 alpha, labeled here as ERN1. That's just the gene name. So we were very happy to see we're getting selective degradation there as well. And with that, I would just like to thank everybody at CPD, especially those at the top of the list who were able to contribute to this work. And with that, I will take any questions.
Great job, Brianna. Thank you very much. So I'm sure the audience has some questions. Let's take a look. Yeah, so how generalizable is this approach given the size of the combined DTAG spycatcher HiBit construct? Have you had examples where the tag interferes with the endogenous protein function?
No, we haven't had any examples of that yet. I would say the clones are a little bit trickier to get, but it's pretty close to what you would see with just HiBit.
Okay. So the linker is very short on the IRE1 alpha degrader. Do you think cooperativity plays a big part in the activity of this particular degrader in measured versus predicted ternary complex formation?
Yeah, that's a great question. We haven't measured cooperativity in this case yet, but based off of our modeling, I would definitely say there is a role for cooperativity. We just have yet to actually measure it, so I wouldn't be able to say that for sure.
Another one. Did you explore any? So the IRE1 alpha ligand is covalent. Did you explore any non-covalent options for this target?
No, not in this case.
Okay. And we'll go with this last one. Are there plans to commercialize the HiBit SpyTag system similar to Promega's collection of traditional HiBit?
No, we've released in our publication, we have the sequence out there, but we're not planning on commercializing it ourselves now.
So it's just available as a tool. Fantastic.
Mm-hmm.
So with that, I'd like to thank you again, Brianna, for a fantastic talk. And we'll move along to our penultimate speaker for today's session, Basil Karim. Basil is a rising fourth-year graduate student in Nathaniel Gray's lab. Before joining the Gray lab, he completed his bachelor's in chemistry and biochemistry and molecular biology at Pennsylvania State University. There he trained under Dr. Jin Jiang, where he developed solvatochromic dyes for protein aggregation detection. He's now enrolled in a PhD program at Stanford University, where he's developing a variety of projects involving PROTACs, molecular glues, and other proximity-inducing bifunctional molecules. So Baz, the floor is yours.
Kind introduction. So as I mentioned, my name is Baz. I'm a third-year graduate student in Nathaniel Gray's lab at Stanford University, and I'm delighted to give my presentation titled Borrowing Transcriptional Kinases to Activate Apoptosis. As many of us know, there's often an imbalance between aberrant proliferation and the evasion of apoptosis in cancer. And many of the drivers of these aberrant proliferation pathways are actually very well-known kinases, such as BRAF, BCR-ABL, EGFR, and others. And these are often dysregulated due to mutations, translocations, and/or overexpressions in certain cancer subtypes. Simultaneously, cancer also utilizes these dysregulation mechanisms to evade cell death. And many of the ways that cancer does this is through transcription factors that regulate some sort of apoptosis pathway or growth arrest pathway. The activity of the drivers of proliferation and regulators of cell death, the drug development community has developed selective inhibitors that target a druggable pocket within the protein. And then more recently, like many of the talks today, have developed degraders or PROTACs to induce proximity between an E3 ligase. And this obviously, as we know, results in the degradation of the protein. And there are many examples that are now entering phase 1, phase 2 clinical trials. More broadly, however, these compounds are examples of what are known as occupancy-driven pharmacology and event-driven pharmacology paradigms. And these are distinct mechanisms where we try to degrade a protein so that we can lose the function of that protein. So they're loss of function strategies. However, given the coexistence of both of these pathways, which are sometimes regulated on a transcriptional basis, we then asked ourselves, can we rewire the transcriptional state of cancer cells to reactivate apoptotic gene regulation through a bivalent small molecule? Turn the strategy of shutting down aberrant activity of either an oncogenic driver or a transcription factor to harnessing them for targeted therapy. And so in this project, we're going to talk about a new gain of function event-driven pharmacology strategy that rewires cancer drivers. And specifically, I'm going to be talking about kinases to activate cell death genes that are turned off. And so to develop this concept, and it's highlighted here, we chose to focus on the BCL6 protein in diffuse large B-cell lymphoma, or DLBCL. Why? Is that BCL6 is mutated or dysregulated in about 60% of DLBCLs, and it represents a strict dependency for these cancer cells because it suppresses the transcription of downstream death genes. It does so by binding a specific DNA sequence with its zinc finger, and then mediating protein-protein interactions with this BTB domain. And it actually recruits co-repressors such as NCOR, BCOR, and SMRT. And so having known about BCL6 for over 20 years, there's obviously been a lot of medicinal chemistry efforts to try and drug it through primarily this BTB interaction domain. To some of the previous work that this project utilizes. As I mentioned, there's a lot of medicinal chemistry efforts that have yielded very selective probes to target the BTB domain of BCL6. Just to point out a few, there are reversible inhibitors such as this compound BI3812. There's a covalent inhibitor that actually the Gray Lab found, which is TMX2164. And there's also molecular glue degraders such as CCT and BI3802, as well as PROTACs. However, unfortunately, up until recently, with the discovery of the Arvinus degrader, most of these BCL6 BTB inhibitors and molecular glue degraders exhibit only modest anti-proliferative effects in lymphoma. And this is primarily due to a loss of function strategy. You need to have close to 90 to 100% of degradation in cells to really get a therapeutic effect. Inhibition or degradation, we chose to leverage the BCL6 protein by developing transcriptional chemical inducers of proximity, or TCIPs. And essentially, these molecules link BCL6 inhibitors to binders of transcriptional activators to induce proximity. This allows for then the repressive functions of BCL6 to be forcibly overridden by the proximity of an activator that then recruits transcriptional machinery to transcribe BCL6 controlled genes. And then the first iteration of this project was recruiting BRD4, which is an epigenetic reader protein to activate BCL6 genes. And we did this by synthesizing a compound called JWZ77, which is otherwise known as TCIP1. And this compound potently kills DLBCL cell lines at about 1 nanomolar through a fundamentally different mechanism than inhibition or degradation. Instead, this compound relocalizes BRD4 on chromatin, which then leads to the transcription of BCL6 target genes and induces a very potent cell death in DLBCL cell lines. And so given that BRD4 is part of this super enhancer promoter complex and helps coordinate RNA Pol2 activity, we then asked ourselves, can we extend this to kinases? And this would essentially make our design modular, and we could, essentially try to recruit kinases to activate transcriptional processes. And so this is kind of the figure that here we're looking at. And so we chose to focus on cyclin-dependent kinases, or CDKs, because they are typically known to interact with transcriptional machinery. And in specific, we chose to focus on recruiting CDK9 and the PTEF-B to BCL6, as this protein is directly known to phosphorylate RNA Pol2 to induce transcription elongation. And so this would allow us to, again, expand our TCIP technology and also utilize the variety of well-validated ligands that exist for targeting kinases. Perfect combination of ligands, we embarked on a very extensive medicinal chemistry campaign, which generated over 100 bivalent compounds using ATP competitive CDK9 inhibitors linked to BCL6 ligands. And then we evaluated them in a drug cascade, where we first tested them in a reporter assay that essentially reports on BCL6 controlled genes, a ternary complex assay using purified protein, and then a cell killing assay to evaluate potency. Through our lead compound here, which is CDK-TSIP1. And so CDK-TSIP1 originally was BACO421, comprises the CDK9 inhibitor SNS032. This is shown here in this green color, linked by an alkyl C6 linker to the BCL6 inhibitor BI3812. And additionally, we synthesized two negative controls, being 0454, which is essentially neg1, and it has this dimethyl bump on the BCL6 inhibitor, as well as the inactive kinase binder containing the methylated hinge interactor, which is known as neg2, also known as 0466. How do I get, there we go. As I just mentioned, we see that CDK-TSIP1 robustly activates BCL6-driven GFP expression, whereas the negative controls do not. Complex assay, we see that CDK-TSIP1 induces a very potent, or a very pronounced ternary complex between purified CDK9 and BCL6 with the characteristic hook effect. And lastly, in our cell killing viability assay, we see that CDK-TSIP1 kills DLBCL cells at about 8.6 nanomolar, whereas the inhibitors in a 1-to-1 molar concentration that are not connected by a C6 linker are about 250 times to 300 times less potent, and the negative controls are about 10,000 times less potent. Evaluate this, whether or not whether or not our TCIPs are comparable to CDK9 inhibitors and degraders, because this is known to be very toxic to cells. And what we can see in SUDHL5 DLBCL cells, CDK-TSIP1 is just as potent as SNS032 and a clinical compound developed by Novartis, NBP2, and considerably more potent than thal SNS032, which is a cereblon-based degrader. Chose to evaluate specificity to ensure that our drug has a therapeutic window. And since BCL6 dysregulation is a hallmark of DLBCL, CDK-TSIP1 really shouldn't be active in a non-cancer. And so to do so, we obtained primary lymphocytes as a healthy cell model. And we observed that these cells were 1,000 times less potent to CDK-TSIP1, whereas the drugs targeting CDK9 retain their toxicity. And so this shows that at least in vitro, our CDK-TSIP approach can potently and specifically target cancer cells with minimal on-target toxicity in vitro. For a couple key proteins that BCL6 regulates, and just with 30 nanomolars of the CDK-TSIP1, we see an increase in expression of pro-apoptotic proteins such as P53, PUMA, and NOXA. When we do RNA-seq, we see that there's a dose-dependent increase in transcription. And when you annotate these transcripts, they correspond to the same pro-apoptotic and cell cycle arrest protein seen by Western blot. Studies, we found that CDK-TSIP1 caused rapid recruitment of CDK9 to known BCL6 binding sites on chromatin, whereas other genes that do not have this binding site are largely unmodified. And upon further investigation at BCL6 bound loci, we observed rapid recruitment to CDK9 as indicated by the 2-hour trace, and increased RNA Pol2 throughout the gene, indicating that there's induced transcription. We wanted to evaluate these compounds in vivo, and unfortunately with the unoptimized linker of CDK-TSIP1, we needed to go back to the drawing board and improve the metabolic properties, and so came up with CDK-TSIP2, which is known as 04232. CDK-TSIP2 is about 10 times more potent than CDK-TSIP1 as indicated in the viability curve by this blue line here. And it also has increased GFP reporter activation and has about a 3-hour half-life, which obviously isn't permeable or permissive for clinical trials, but at least for a tool compound, it is permissible. And so lastly, reiterate that BCL6 is a master regulator of cell death. But the endogenous function of BCL6 is that it helps promote B cell maturation processes. And so B cells in the germinal center will upregulate BCL6 so that they can essentially not die during these maturation processes. And when we add our drug in an humanized mouse model, we see that there's a dose-dependent decrease in germinal center B cells, which are high expressing BCL6 cells. However, naive B cells and memory B cells are largely untouched. And this is really showing how that our compound is targeted just for BCL6, whereas other B cell targeting therapies such as immunotherapy would essentially wipe out all these types of cells. Today, I was able to show that this is a general strategy to rewire transcription. We really focused on the induction of apoptotic genes in lymphoma, but in a broader sense, I just want to say that we are modulating transcription in a context and lineage-specific manner without any genomic alteration, just with a small molecule. And so not only is this a modular side on the activator side, but we also believe it's modular on the repressive side. And so looking forward to the potential applications of this technology, we believe we can explore new therapeutic strategies for a variety of different diseases that are not limited to lymphoma. And we believe that this opens up possibilities for targeted gene regulation that maximizes therapeutic efficacy through a gain of function. And with this, I'd really like to thank Sai Gauri-Sankar, as well as Roman Surat, who are co-leads on this project, as well as many others, such as Saban Nettles, who did the ChIP-seq experiments, Brendan Dwyer, who did proteomics on this. I didn't show that in this presentation, but it was a beautiful experiment, as well as Yuste, Hind, Andre, and Jason, who helped on the cellular biology and biochemistry. And with that, I would like to open the floor to any questions.
Thanks, Baz. That's a great talk. Yeah, we have some time for a few more questions. So cooperativity and kinetic factors such as residence time are important for TPD approaches. How do you view these as important for your TSIPs?
Unfortunately, we haven't done any cooperativity experiments. I think with our BRD4 paper that was published in Nature, we have done those, and it does show to be very important. We clearly see a dependency and a correlation with ternary complex formation, but I can't comment exactly on how cooperativity affects TSIPs without the data there.
Yeah, sure. Yeah, next question. Have you conducted any analysis of the CDK TSIP induced phosphorylation pattern, or perhaps a zone of phosphorylation around the target?
So One experiment that we did is that we made these FKBP CDK9 conjugates in SUDHL5 cells, and we infected them into the reporter. And we did find that there was a requirement for recruited CDK9 through FKBP, essentially an active kinase domain. And so if you inactivate the kinase domain, it doesn't activate GFP expression. We also did phosphoproteomics, and those results are a little bit less clear, but we're following up on them.
Oh, cool. Okay. The RNA pol-2 ChIP-seq data is particularly convincing. Has there been any characterization of broader histone marks or higher order chromatin structures after treatment, particularly at BCL6 sites with more heterochromatin?
We haven't done those experiments yet, but we are definitely following up on them. Yeah.
And I'll do the last question before we move on to the final speaker. So as a technique or generalizable approach, how adaptable could this TCIP approach be towards other applications, for example, stem cell or cell reprogramming work?
I think it's going to be very generalizable. However, I think what most of this audience knows is that you need a good ligand for transcription factors, and that is no easy target to have. So I think with the coming years, when we develop more ligands and more medicinal chemistry to get good binders, this can absolutely be an approach for other diseases and other applications. Yeah.
No, fantastic. Okay. We have a few more questions in the chat, Baz, and we'll assign those across to you now to answer if that's okay. And yeah, thanks again for a fantastic talk. And with that, it's time for our last speaker in today's session. So Scott Hughes. Scott is a scientific co-founder of the University of Dundee spin-out Amphista Therapeutics, where he is currently the director of exploratory biology. He completed his PhD in pharmacology at the University of Toronto and then joined Alessio Ciuli's group in 2016 to work in the field of targeted protein degradation, focusing on structure-based PROTAC design and the characterization of novel PROTAC-induced ternary complexes. So with that, I'd like to hand over to you, Scott. Looking forward to your talk. Thank you very much.
Great, thanks very much for the introduction, and thanks very much to the organizing committee for giving me this opportunity to share a slice of a story from Amphista that's near and dear to me, titled Degradation of BRD9 by a Novel Targeted Glue. And actually, thank you also to all of the speakers who've given some fascinating, interesting talks and also done a good job of introducing TPD and a lot of the technologies that I'll be mentioning here as well. So, Amphista was set up to expand the landscape of bifunctional degraders beyond the usual suspects, so your Cereblon and your VHL, to really deliver innovative medicines to patients with unmet medical need. And how we go about achieving this is by identifying novel chemical matter that drives the selective degradation via target-induced E3 ligase interactions. This is done by designing chemical libraries based on drug-like compounds with good properties in vivo and CNS penetration. Our degraders have are able to recruit multiple E3 ligases depending on the nature of the target. And through this approach, we've been successful in integrating a diverse set of target classes in different subcellular compartments. So our discovery pipeline employs a sort of degradation first approach. So whereby we, starting with a ligand for a POI, a protein of interest, this is then derivatized at a solvent exposed region to generate, using our proprietary library, to generate a library of bifunctional molecules. These are then screened using a high throughput degradation assay, typically a high-bit assay. And then following successful degradation, this is confirmed using all of your appropriate controls before finally then moving on to identify and characterize the mode of action, which E3 ligase is involved, etc. And really only the well-characterized, well-refined mechanisms, clean mechanisms are taken forward. The compounds are moved forward from there. So today I'm going to tell you about a very specific case around BRD9 and take you through this process from conception through to the mode of action. So BRD9 is an emerging target in oncology. It's been explored for therapies in synovial sarcoma, malignant rhabdoid tumors, as well as leukemia. It's actually a really good target for TPD. And this is mainly because BRD9 inhibition causes only modest phenotypic effects. And in this regard, degradation is not equivalent to inhibition. You do get very different pharmacological responses when degrading the target. And fortunately, you also have a range of high-quality ligands available, which have these nice solvent-exposed handles. So starting with this POI ligand, down here, we derivatize this solvent-exposed region using our library of linker warheads, and then screened for activity using a classic high-bit BRD9 HEC293 cell line. And here we identified novel sub-nanomolar degraders of BRD9. I've shown here one example, compound 1, which has a very nice sub-nanomolar DC50. Just to orient you here, in pink, we've got the POI ligand, the linker in the middle, and then over here in sort of blue color is your cyanoacrylamide warhead. Interestingly, this screen also identified a matched pair negative control, shown here as compound 2, which showed no activity or degradation. So we were quite pleased to see this because it already from the beginning suggests that there's a well-defined SAR in these molecules. And the fact that the only difference is the loss of this removal of the cyano on the cyanoacrylamide suggests that there is an importance of that cyanoacrylamide moiety, which keep that in mind for the future points. So looking for degradation activity in HEC293 is all well and good, but of course we were interested to know if this extended to cancer cell lines, both solid and liquid. So to do this, we pivoted away from the high-bit to a immunofluorescent assay. It's still high throughput, but it is cell agnostic, and there's no tag system, so it allows us to profile a lot more cell lines with ease. So I've shown two examples here where if you treat compound 1 for 6 hours in a representative AML cell line, MB411, a representative breast cancer solid tumor line, MCF7, you're still getting very nice nanomolar or sub-nanomolar degradation and a good Tmax value. So in the selectivity front, we then went on to perform global expression proteomics in MB411 cells. So here we took compound 1 and treated MB411 cells with 100-fold over the DC50 for 6 hours, and then looked at the selectivity by proteomics. We're very pleased to see we've got very nice selective degradation of the target of interest, BRD9, and by contrast, you're seeing negligible degradation of BRD7, which is a compound or protein that the POI ligand also engages, as well as no degradation of BRD4, which is a known and unwanted off-target of these compounds. So satisfied with the potency and selectivity, we then moved on to understanding the mode of action. So to do this, we started like you would with any typical PROTAC or bifunctional molecule in looking at the effect of a range of cellular compounds. So when treating with your classic MLN4924 or bortezomib, you see nice complete blockade of degradation by compound 1, showing that these compounds are metallation and proteasome dependent, and thus likely dependent on a colon ring E3 ligase. Similarly, as expected, treating with excess POI ligand, so the BRD9 POI ligand is able to block degradation by compound 1. So all good here. But then, interestingly, when we went to do the reverse of that blockade and look at excess treatment with the cyanoacrylamide warhead, we were surprised initially to see that it is unable to block degradation by compound 1. So this was the first piece of evidence to suggest that maybe these aren't quite the bifunctional PROTACs we may have thought they were, and maybe a bit more glue-like behavior is coming out. So from this, we were really quite curious to understand what E3 ligase was involved in that. So to do that, we revisited the high-bit BRD9 cell lines and performed some ternary complex pull-down experiments. So here you've got your high-bit tag BRD9 cell, and you treat with either compound 1 or the negative control compound 2, and then lyse the cells and do a pull-down with anti-high-bit, and then analyze the outcome by proteomics. And quite clear with compound 1, we see very nice strong enrichment of DCAF16, which is a E3 ligase receptor, and also DDB1, which is a known, is the adapter protein for DCAFs, and specifically DCAF16 in this case. So we were quite well pleased with that. And also, to further convince us of the specificity of this interaction, the enrichment is dose dependent. So at higher concentrations, you see more enrichment of DCAF16 and DDB1. And by contrast with the negative control compound, so the compound missing and lacking that cyano group, shows no enrichment of DCAF16. So this is very interesting because this is again where we're seeing this, this difference with the negative control, and we want to try to understand, what is it about the cyano group? So I may have not mentioned it so much in the beginning, but cyanoacrylamides are thought to be reversibly covalent. There's quite a well-bodied literature to show that they engage cysteine specifically. So we wanted to investigate this a little bit further, and to do so, we looked at intact mass spec and recombinant protein. So we recombinantly expressed DCAF16 and DDB1, as well as the bromol domain from BRD9, and then performed a few intact mass spec experiments. So in the top here, if you just take DCAF16 protein alone and incubate with 5 equivalents of compound 1, and then allow the reaction to reach equilibrium, and then measure the adduct formation using mass spec, you do see adduct formed that is detectable, but it is not the dominant species. So the majority of species detected is actually the unbound, which is maybe not fully expected given the molar excess of compound. By contrast, if you incubate in the presence of BRD9, so just one equivalent here, so 1 equivalent DCAF16, 1 BRD9, and 5 equivalents of compound 1. In this case, when you measure adduct formation, you see that the dominant species is actually the labeled form, which really does suggest that BRD9 is actually catalyzing the formation of this adduct and is shifting the equilibrium in favor of the modified, which is the degradation competent species. So obviously, for this adduct to form, there must be a specific cysteine which is engaged. So we took these incubated samples here and did some peptide mapping mass spec and identified cysteine 58 as the engaged target on DCAF16. So this is interesting. It's one of 3 accessible cysteines on the surface of DCAF16, these others being 173 and 178. And what's interesting with 58 is that it was also identified as a target of a BRD4 degrader, which was characterized by Eric Fisher's group. So all this, of course, has been done with recombinant protein, but we very much wanted to confirm that this exists at a functional level. So we generated some cysteine 58 mutant knock-in cell lines in MCF7. And here you can clearly see that, whereas in red, you've got your degradation with compound 1, you're losing that in both the complete absence of DCAF16, but also by simply mutating that cysteine to a serine. You basically recapitulate the knockout activity. So this had us fully convinced, of course, that our mechanism was dependent on cysteine 58 of DCAF16. So the chemists in the audience, if there are I'm sure they're out there, may have noticed that the warhead here actually contains a stereocenter. And actually all of the data up till now has just been with the racemic mixture. But we wanted to understand the importance of this stereochemistry, and so we separated the 2 enantiomers and tested them for activity, and we're very pleased to see you actually have a difference, a very noticeable difference between the 2 enantiomers. So enantiomer 1 in this case gave much greater degradation than the other enantiomer. And this actually correlated very well when we went to look at ternary complex formation. So this was done here using, again, with recombinant protein, using a sort of classic alpha-lysa proximity assay, where you're looking at the amount of complex formed in the presence of increasing amounts of complex. And here you see an enantiomer that gives you greater degradation also gives you much more efficient ternary complex formation. So this was a very pleasing result because it really suggests that rather than just a gob of reactive glue stuck on the end here, we've actually got a very, the warhead stereochemistry is actually very important in driving the molecular recognition. It's not merely the reactivity of the compound. So if we bring this all together now, we've sort of landed at a sort of proposed model for how our targeted glue molecules work. And it functions by this sort of template-assisted modification of DCAF16. So when the compound is first introduced, it's going to bind preferentially to BRD9, and this is by virtue of the kind of nanomolar affinity for BRD9. You may have some modification of DCAF16, but it's probably reversible, and we're not sure quite about the level of stability there. Once bound to BRD9, this will then form a ternary complex with DCAF16, stabilized probably by some protein-protein interactions. And this brings the warhead, so the cyanocrylamide warhead, into close proximity to the cysteine 58 and holds it there long enough to allow for that covalent modification to occur. And it's very likely that the different enantiomers are actually accommodated differently within this ternary complex, which accounts for the differences in activity that we see. Once this ternary complex is, of course, formed and we have the adduct formation, then within the context of the larger ubiquitin machinery, you'll have ubiquitination and subsequent degradation of BRD9. Now, this adduct then is retained on DCAF16. It's primed to allow for binding of subsequent proteins of BRD9 and subsequent rounds of degradation in a sort of degradation cycle. So I did mention at the beginning that, of course, we're very much interested in producing compounds with drug-like properties. So we wanted to go on to look at how these behave in vivo. So when you dose the enantiomers, both of them actually, so I haven't actually put the dose on here, but it's 10 mg per kg oral. If you dose that, you do get nice bioavailability with both enantiomers, 20 and 30% respectively, which was very encouraging to see. And then when we go and look at this in an MB411 xenograft model, so here we're again dosing orally and then looking for degradation of BRD9 in the tumor samples. And here you can see that there is a very noticeable level of degradation with the one enantiomer versus the other one. And this, of course, does match with our in vitro data as well as the alpha-lysa data. So here we've demonstrated in vivo activity of a reversibly covalent targeted glue that recruits DCAF16. So to summarize, hopefully you've seen now and convinced that we've demonstrated potent selective drug-like degraders of BRD9, discovered using Ampista's proprietary chemistry library, and these possess in vivo activity after oral dosing. So DCAF16 is selectively recruited to BRD9 using this covalent targeted glue. And these, as far as our understanding of it, these rely on specific molecular recognition within the ternary complex based on the stereochemistry as well as the reactivity within the warhead. And although I've not shown it here, we have used this approach to, degrade multiple different targets and also retaining drug-like properties. And in some cases, we've been able to achieve this using different E3 ligases from those I've shown you today. So probably the most important slide is the acknowledgements and thank you. So this has been quite a journey at Ampista. And I'd like to point out, in particular here, of course, Andrea and Wojciech or Jay have been quite instrumental in a lot of this work shown here. Colin did a lot of the proteomics work as well. and everyone else has been involved in varying degrees as well. So thanks again for listening and for your attention, and happy to take any questions.
Thanks, Scott, for a really good talk. Great to see some new data from Unvista and a new story. Yeah, lucky for you, we have plenty of questions, so we'll get straight onto it. So very inspiring talk. Could you comment on the effect of trans and cis conformation in compound 1 covalent warhead?
Good question. The trans and cis in terms of the cyanoacrylamide. So I can't actually comment specifically on those aspects of the chemistry. It's something we have considered in terms of it, but actually in terms of measuring the specific contribution of it, I can't comment specifically on the data.
Did you look at proteome-wide cysteine labeling, and is that a safety concern with covalent compounds?
So we have looked at that in some capacity, and actually you do see very few modified cysteines compared to other acrylamide, or certainly some of the more reactive chlorocetamide. But I would caveat that we have still encountered some technical issues because these are reversibly covalent. So there are some challenges in interpreting the data in that capacity. But we generally believe they are quite selective in their engagement.
Okay, thanks. Have you observed the glue-like behavior using other decaf-16 binders? E.g., is it specific to the decaf-16 binder that switches from PROTAC to glue-like behavior, or is it likely something more general about BRD9 and decaf-16 specifically?
So I can't comment on specific targets of E3 other than we have observed this with other targets and other E3 ligases. Yes, to that.
Regarding your mechanism of degradation and complexes, your explanation would also accommodate your inactive irreversible acrylamide. How do you rationalize this at present?
Yeah, so I think there is probably a recognition component of the cyano, but I think also the inability to form that adduct and the covalent modification is probably a key driver in the lack of activity.
So a more, I guess, general question. I guess in your opinion, what opportunities are presented by recruiting decaf-16 over the standard recruited ligases, cereblon and VHL?
I think different targets will have a different capacity for being degraded, and I think it really comes down to the chemical matter ultimately. I mean, I think there are probably properties of E3 decaf-16 versus cereblon versus VHL in terms of expression profile and their capacity for certain targets. But I think it's at the core, when you're trying to make a drug-like compound, it's really about the options from a chemical perspective. I think is where that it's nice to have that broader toolbox.
Yeah. Okay. No, that's good to know. And maybe we'll call this the last one. We can stop your Viva-like Q&A session. Do you think that after creating covalent bond with decaf-16, your warhead can degrade other targets by changing the selectivity?
Well, I mean, our global proteomics at 6, and we've also run it out to 24, and you don't really see much of a change in the degradation selectivity. I mean, obviously, once you get to a longer enough time point, you start to get secondary effects, which will probably come in there. But we haven't seen any evidence that you would have any off targets coming in over a period of time.
Thank you. Yeah, I'll leave it there, Scott. But thank you so much once again for joining us. And I guess with that, we'll close out today's event. So thank you all to the audience for joining us for the 5th installment of our annual TPD and Induced Proximity Symposium. As we wrap up, I'd like to mention that we have a short 5-question survey immediately following the event. So if you have a few moments, we'd love to hear your feedback on this event, as well as what topics you're interested in for future symposiums that we will put on. And now it's time to end. So it's been our pleasure to bring you this event, and I want to thank all of our wonderful speakers for the incredible research shared and time spent preparing for the event. I'd also like to thank you all in the audience for all over the world joining us today. I hope you have a wonderful day, and we look forward to seeing you at future events and webinars. Thank you very much.